Okay, let's go ahead and get started with this third part to this chapter 3. Yeah, okay. So sorry about this last section had the audio didn't start right away. So for whatever reason, it started mine as muted, and I was just talking away, and it didn't record that. So anyway, I think I got that squared away this time. All right, so 3.6, carrier-mediated vesicular transport assist membrane passage of specific substances. Okay, so this is just describing the process for which things will pass through the membrane of the cell. So carrier-mediated just means it's a receptor site, or the thing that's allowing it to pass through is mediated. This is a passive process, which is to say that it does not use ATP. Okay, all right. So what is carrier mediated? Well, number one, it's very specific. It's like an enzyme. Remember, proteins, it's all about the shape. Remember, proteins, it's all about the shape. So each carrier mediated transport system is very specific. For example, like to allow glucose to enter the system or the cell. It can only go as fast as its limits are. In other words, it can only go as fast as there is stuff outside the cell, or it can only go as fast as there is stuff in the cell membrane or the plasma membrane that allow that stuff to pass in, right? So it's what we call saturation limits. And of course, regulations, just like an enzyme activity, often depends on presence of cofactors. The binding of other molecules, such as hormones, can affect the activity of carrier proteins. For this reason, hormones provide an important means of coordinating carrier protein activity throughout the body. Okay. So facilitated diffusion. This just means that things are allowed to pass in. So facilitated diffusion, we can have something like glucose out here. All right, this is a ligand out here. And this glucose, remember, glucose C6H12O6, right, has a shape to it. So glucose comes, it's not a protein, it's a sugar, but it still has a shape to its organic chemistry, right? Well, it comes into this protein, which does have a shape to it, and this is an integral protein. And this glucose then attaches to this receptor site in this protein. And if I put a bunch of molecules, which we already talked about as in positive, negative charges, whatever, it's a shape to it. If I put that on the protein, it changes its shape. And the changing of the shape then allows it to open up on the inside. Once you change the shape, this receptor site no longer can hold this glucose molecule, and it drops it inside the cell. Okay, so now you can see that the saturation of this is either determined by how much is out here in the glucose or how many of these you have. This can only go as fast as it can go. So one will come in, attach, release, and drop it in. If there's nothing out here, maybe one passes by occasionally, boom, it drops it in. So that is saturation limits depending on what it is. And this is a passive process. It does not require energy for this to occur. All right? It does not require energy for this to occur. All right, so active transport systems are just that. They are actively transported across the membrane. Ions like sodium, potassium, calcium, magnesium, these all, remember we talked before that sodium likes to live outside the cell and potassium likes to live inside the cell. And when we open proteins, integral proteins, or potential voltage gated ion channels, like the sodium will pour inside the cell, okay, or the calcium will pour inside the cell, or potassium will leave the cell, right? So those are active transport systems, and these are called ion pumps. Of course, those are ions, right? Cations, right? And these are exchange. So we have some exchange pumps here. Okay, so we have a sodium potassium exchange pump. So this is an ion pump, just pumps the ions, but then we can have what's called an active, primary active transport, or a sodium potassium exchange pump. We have these all over our body. So sodium's like on the outside, potassium's on the inside. Okay, and now what happens is the sodium will come into the cell and potassium will leave the cell. Well, once that process is done of those changing, you have to get them back, right? And to do that, you actually have to pump them. This is like, okay, so coming down or diffusing down, what they normally do is sodium will diffuse into the cell, potassium will diffuse out of the cell. Remember, it's net movement of particles down the concentration gradient. So to get them back to where they go, you have to, it's like pumping water upstream, right? It's not going to just flow there, so I actually have to pump it there. So it doesn't matter which way the water is moving, a pump can put it either downstream or upstream. Okay, it doesn't matter. Okay, all right, so that is, here it is. This is showing you sodium being pumped out. Remember I said sodium likes to live outside, potassium inside, and then sodium pours in, potassium out, and then now we got to swap it. And of course this uses ATP, right? So got to put it back to where it was originally. So we have that voltage difference. So this is, remember, this is just like, this is potential energy. When you dump it in, that's kinetic, and when you store it, it's potential energy. Okay, all right. Secondary active transports just show that another one is occurring as a result of this process. So you see that sodium attaches, and then glucose molecules attach, and then it drops off, and so on and so forth. Okay, so it's not a super big, it's not a big deal. Just kind of take it for a second. It's just a secondary. It's another way to get glucose in at the same time as when sodium's coming in. Okay. Now remember we talked about vesicular transport and vesicles, and vesicles are, what are vesicles? They're pouches, right? So vesicular transport is an active process. It does require the use of energy, ATP, right? So we talked about vesicular transport as when the rough ER takes those modified and packaged newly synthesized proteins and ships them to where? So it shifts them through vesicular transport, which is a passive or active process, and it sends it to the Golgi apparatus. Okay, now that Golgi apparatus then modifies it some more, packages, stores it, gets it ready to leave, and it leaves the cell through a process called exocytosis. Okay, so that's that protein synthesis, transcription, translation. You can see that on that spotlight for transcription and translation. Okay, so vesicular transport, active process. Endocytosis means it's coming into the cell. All right, we have a couple of those. We have receptor-mediated endocytosis, which is where just a receptor controls the process when it comes in. Penocytosis, where we have, this is cellular drinking, and phagocytosis, which is cellular eating, cellular eating. All right, and this would be like a white blood cell eating or engulfing a bacterium, okay, a pathogen, some damaged tissue, whatever, okay? And they do require the use of ATP, which we get from where? Right, the mitochondria, which produces what? ATP, what's the cycle? A Krebs cycle, tricarboxylic acid cycle, citric acid cycle, right? And those are aerobic respiration, right? Okay. Okay. Okay, this is just showing you receptor mediated endocytosis. Here it's coming in endocytosis and then exocytosis. This is just a plasma membrane out here, this line. And you see that these things are out here, hits receptors, it starts to invaginate, makes a vesicle, right? Then this vesicle, then the primary lysosome will come over, attach to it, do its thing, and then of course it puts the plasma membrane back on the surface over there, okay? A nice chart to overview all of our transport systems that were passive and our active processes there. Our passive and our active processes there. A transcytosis is just talking about as the material comes in, for example, into your capillary beds, into your digestive tract, as something comes up to the one on the inside or the luminal side of whatever, this material will invaginate. It'll come through the cell in endocytosis. It gets to the other side of the cell. Like for example, if this box was a cell, it would come in here, it would attach here, it would come inside the cell through endocytosis, then it would go down and it would hit the other side of the cell, and then exocytosis would let it go out. Right, so it'd come in through a vesicle, would cross over, and then it would release it. So endocytosis coming in and exocytosis leaving. Pretty straightforward. So go ahead and check next section here. Okay, so this is just talking about the potential difference in membrane potential, which is, I kind of alluded to this before, sodium on the outside of the cell, potassium on the inside of the cell. We open up these sodium channels, and then each of our tissues have what we call resting membrane potential. For example, cardiac muscle tissue rests at minus 90 millivolts. Okay, now the further away from zero it is, the stronger that potential is. So the further away it is, the faster it pulls in, like for example, sodium. Sodium loves to come into the cardiac muscle cell because it's at minus 90 millivolts. So it pours in really quick, right? So when it pours in, it changes this resting membrane potential, and it starts going up. Once it hits minus 70 millivolts, that cardiac muscle will actually open up voltage gated channels, and it goes through what we call a heartbeat. We'll talk about that when we get into anatomy 2, but just understand that all tissues have a resting membrane potential. Okay, all tissues have a resting membrane potential, and we use the ions coming in or out, those sodium, potassium ions, to do the work. Remember, those ions are actually voltage, right? So if I bring in a positive ion into minus 90 millivolts, what happens to this voltage? It actually goes towards zero. So minus 88, minus 86, minus 85, 84, 32, 70. Once it hits minus 70 millivolts in cardiac muscle tissue, voltage gated channels open up, sodium pours in, and it causes a heartbeat. All right, what we call depolarization and repolarization, which we'll get into that in anatomy 2. Okay. So those ions are how we do work and everything. All the things that happen in our body are based off of ions. Okay, so mitosis. Okay, we're not going to get crazy with mitosis. All right, just understand that mitosis is the division of our 75 trillion cells in our body. That's a lot of cells. Cellular division. So when we split up one cell and make it into 2 cells, they're called daughter cells. That is the process of somatic cellular division, is what we're going to talk about. Okay. Genetically controlled cell death. This is just definition. So cells they're predetermined when they're going to die. That is called apoptosis. All right. That is genetically controlled cell death. That's a definition for you. Okay, so here's the cell life. This is more than enough information for you guys for mitosis. So make sure you understand the wheel of the cell's life cycle. Put a couple words to describe each one of these sections. Here is mitosis right here. This is a normal cell's life. This is mitosis. So you see it starts with PMAT, prophase, metaphase, anaphase, telophase. This is mitosis, and it ends after cytokinesis. Okay, so that's that right there. So these guys right here are this right here. Okay, so as it comes in, cells start to get ready for mitosis through these phases here. This is the gap phases. This is normal cell life cycle, which you can even put that, just to describe it. You read these guys down here, these little descriptions. You see, we talked before that this chromatin starts to coil up, right? Centrioles go to one side, they polarize. One goes up, one goes down. The nuclear envelope starts to dissolve. Chromosomes are all nice and tight now, or the DNA is nice and tight and wrapped up around those histones like we talked before. We end up with this line. It's called a metaphase plate. The centrioles release those microtubules. Remember, those are the filaments, microtubules, which come down attached to either side of the DNA. Pull one half of the copied DNA one way, the other the other. We get a cleavage furrow, and that cell then separates, creating daughter cells. Okay, so go through that, read and understand. We can actually see this in the lab. We can see, in our lab, we can actually see this process occurring right here. Okay. I think you guys have pictures of that in your lab book too. So they look just like that. Okay. All right, so we have interphase and DNA replication. It goes through this. This will help you read this. Listen, you do not have to go through DNA replication. Okay, you do not have to do DNA replication. You do need to understand that we make a copy of the DNA. We make a copy of it, and then one half the DNA goes to one side, the other half goes to the other side. They are identical matches because of the complementary base pairs, and then how they make copies of each other that way, okay? All right. All right. Okay, so let's go ahead and move on. Okay, cells. We're not going to get into what causes a cells to go through, what change, causes them to go through the change or the mitosis. But if you come down here, there is something here, telomeres. Now, telomeres are the ends of the strands of DNA, and they do, if they start to fray, this is where we start getting changes or problems with DNA replication. If they start to fray, this keeps the caps. This is like the ends of your shoelaces. If you have those hard plastic things, well, they kind of keep them. They keep them so they don't unwind. Okay. All right, so let's go through a couple definitions. A tumor is a neoplasm, which is just a mass or swelling produced by abnormal cell growth. Most of the time, tumors are encapsulated. If that's a benign tumor, encapsulated rather. So tumors are not necessarily bad when they're benign, except for if they're mechanically in nature, mechanical, where they actually impinge something like nerves or blood vessels, or the location, they're space-occupying tumors. Okay, those would be bad, like in your brain. Malignant tumors now, they're the ones, of course, we all know this, that they're the bad ones. They actually invade tissue and cause them to change, or they're mutagens. So anyway, malignancies are bad because they invade the tissue, invasion, all right? And then those are called primary tumors. They can create secondary tumors where this tissue will go off and you'll have malignancies of, say, kidney cells in your lungs, and that's a secondary tumor. And that's how we know where it came from, is we do the analysis of those cells, and we can see that those are some type of kidney changed cell that is now in your lung, and of course that's not supposed to be there. And that's just one example. There's a gazillion and a half different ones. Okay, so anyways, carcinogens are things that create cancers. Mutagens are agents that cause mutations, are called mutagens. And of course, that's metastasis, primary and secondary metastasis. I'll let you read the rest of that. Make sure you read these through Breakthrough with Stem Cells. Telomeres, Aging and Cancer, that's a great article. Cellular differentiation is cellular specialization as a result of gene activation or repression. So cells change because of differentiation. So something will tell a gene to turn off or on, and that's called cellular differentiation. It's when a cell starts as one thing and then turned on to become something else. Okay, so make sure you read that section on differentiation. All right, so this wraps this up. Okay, this wraps this section up. Make sure that you guys utilize these videos. Make sure that I noticed that the test 1 and test 2 were, I'm sorry, chapter 1 and chapter 2, which was test 1. The grade, the average was a 68.2. I think it was 68.2 or something like that. But anyway, some of you guys are not studying. All right, this is an online class. Unfortunately, it is what it is. You guys, this is an online class and you have to do the work. So make sure you get out there and get your studying done, okay? Okay, anyway, that's it for now. I'll yell at you at another Zoom meeting or something. Anyway, that's it for now. Okay, so I'll go ahead and stop this here real quick.